Thank you.
Alrighty. Next up, we have Dr. Yaqiang Wang. Dr. Wang is an assistant professor in the departments of biophysics and obstetrics and gynecology at the Medical College of Wisconsin. He earned his PhD in biochemistry from the University of North Carolina at Chapel Hill under the mentorship of Dr. Gary Pelock and completed his postdoc with Dr. Julie Fagan at UCLA. He later served as principal scientist at Arrakis Therapeutics, where he led structural biology initiatives in RNA-targeted small molecule drug discovery. Dr. Wang's research centers on uncovering the structural and mechanistic principles of oncogenetic non-coding RNAs and their recognition by proteins and small molecules. His laboratory employs an integrated toolkit to visualize RNA and ribonucleoprotein complexes with the goal of advancing RNA-targeted therapeutic development. His talk today is entitled Isotopic Labeling Strategies for RNA-Targeted Drug Discovery. And without further ado, take it away.
Thank you, Ken. Today I'm going to talk about the isotopic labeling strategies for the RNA-targeted drug discovery. So first, so why we care about the RNA? So RNA actually is a fundamental of all the biology, and so we know the majority of the genome actually transcribed to RNA. And if you look at the transcriptome, 2% of the transcriptome actually coded for protein, 20,000 protein. And around 700 protein is a drug target, already have drug targeted protein. And the majority of protein, especially like a transcription factor, splicing factors, they're just undruggable. So one strategy is to target this undruggable proteins to look at the level, one level above, basically look at the mRNA level. And besides that, with the transcriptome, 98 of the transcriptome is a non-coding RNA. Basically, the RNA is not coded for protein. And from the research we know in the last several decades, we know this kind of RNA is very important. They have a tRNA, microRNA, small interfering RNA, also large and long non-coding RNA like telomerase RNA, group 2 intron. And this non-coding RNAs, they play a very important role in nearly all aspects of cellular biology and are a major contributor to the human disease. So understanding the structure and the function of a non-coding RNA hold a greater potential for the novel drug discovery. And however, to determine their structure is not so easy. So RNA compared to the protein is quite dynamic. RNA have several, various different structure motifs like stem, branch, internal loop, hairpin, and a pseudonode. And if you look at the PDB, so only a little over 2% of the structure in PDB is a nucleic acid. So if you exclude the DNA and also duplicate, and so novel RNA structure is less than 1,000. Compared to the protein, we have over 200K structures. And that's actually the major hurdle for the machine learning like AlphaFold to accurately to predict the RNA structure. So the A3, X3, NMR, and the cryo-EM, they're still like a main three major approach to look at the structure for both the protein and the nucleic acid. But if you compare to the protein and the nucleic acid, and actually the NMR play a quite significant role in the RNA structure determination. That's partly due to the RNA structure is very dynamic. It's very difficult to get a crystal structure, and also most of RNA motif is relatively small. It's too small for the cryo-EM. And besides that, NMR is actually a very powerful tool for RNA-targeted drug discovery. And if you look at from the early stage, the target identification validation, so look at the RNA-QC and the structure validation and the ligand ability analysis. And also move to the screen, NMR have the fragment screen, use the ligand observed and the RNA observed, and also to the head-to-lead optimization to establish its error. And we can use NMR to optimize the potency and the selectivity, and also develop drug-like properties as well. So in today's talk, I'm going to briefly touch, RNA-NMR spectroscopy and also the common approach we use for RNA labeling, like chemical synthesis and in vitro transcription. Also, some a strategy for the large RNA as well. So on the right side, that's just common stable isotope we use for the RNA NMR labeling is a pretty common for the proton, carbon, nitrogen, the fluorine, and the phosphate. So before we move on, so I would like just to make sure we are on the same page. And this is from the textbook. We know the DNA to generate we draw like a two double-strand helix and has ATGC, and RNA is typically draw like a single strand. But actually, the RNA could form a very complicated tertiary structure. So RNA have AUGC, and each nucleotide have three components of phosphate, sugar, and base. And the phosphate forms the phosphate and the sugar forms the backbone over the RNA helix, and the base is usually can form a hydrogen bond with a complementary nucleotide like A to U and G to C. So immunospectrum is the one that I would say is the most frequently used spectrum for the RNA structure, and usually it's kind of serve as a QC to look at how the RNA folded, and so we to look at the proton spectrum, and if you focus on the very downfield shift, typically over 10 ppm. So basically, this region is is usually for the small molecule it's kind of like empty here, but for RNA, each of the RNA if form a base pair like A to U form a base pair, so immuno and will appear as this region from the 10 to 15. And so basically, we can kind of like each of the peak is corresponding to one of the base pair, and also based on the chemical shift, and we can tell and which peak is from the AU base pair and which peak is on the GC base pair, and we also can tell some non-canonical base pair as well. So in order to assign this immunopeak, we typically run it's a 2D nosy, and I just give an example of this very simplified and RNA duplex, the GCUA. And if you look at the 1D immuno, just like it's mentioned, each of the peak is one base pair, so we'll have a 4 peak. So when you collect the nosy spectrum and based on the chemical shift, and we can get the first one is on the very left, that's the first GC base pair, and the first GC will have an NOE peak with the neighboring GC, another GC. So we can find this 2, then 2 will have another NOE with the neighboring 3. So basically, we can go down the road to assign based on their stacking and assign the peak from the nosy. So with this information and then we can look at it a little more on the binding and the conformation response. So on the top panel, that's the 1D immunoproton of our pre-Q1 ribose switch. And again, each of the peak is responding one of the base pair in the pre-Q1. So this pre-Q1 is a kind of response for the calcium, because calcium can stabilize and pre-Q1. And on the bottom is when you add a ligand. So you can see when you add a ligand, a lot of peak change. So it's just a chemical perturbation. And also we see a lot of a new peak. So that indication is once you add a ligand, the ligand will induce a new peak. This peak could from the new from the base pair, and also could be due to the hydrogen bond between the ligand and RNA. So that's a very good indication to suggest this ligand that could cause is our very specific binder. But of course, I want to point out here that so immuno is to provide information on the base pair level. So it cannot provide the binding information from the unstructured region, so like the Borg and the loop. And also for this kind of the big RNA, it's very hard to assign this immunopeak without labeling. So there's a common use is the 2 type of a labeling strategy. One is a chemical synthesis, one is in vitro transcription. So chemical synthesis is very straightforward, and basically you give the sequence to a machine, the machine will synthesize RNA, And this approach will give you quite pure RNA. And also, you can do a specific labeling. Basically, you can tell which position you want to do the carbon or N15 labeling. And you can get a good amount of RNA from this approach as well. And it works very well for the smaller RNA, depends on vendor. Usually it's within 100 to 150 nucleotide. And the only downside is it's quite expensive, and so it's costly. So on the upside, the in vitro transcription, which is the enzymatic synthesis. So basically, you use the T7 polymerase, and just to give the polymerase and the DNA template, and also the AUGC for type of AUGC, and the T7 polymerase will based on the DNA template to synthesize the RNA. So in this approach, and you can just replace the AUGC by uniform like a labeling, or you can just replace only one, like for example, one C is labeled, so you can achieve a nucleotide specific labeling. So for this approach, you can synthesize hundreds nucleotides RNA. It's worked well for the large RNA, and it's really cheap compared to the chemical synthesis. So with this labeling, then we can put another dimension on the immuno to help the assignment. So for here is the proton nitrogen HSQC, and on the you can see the different type of NH appears at different location. On the left lower corner, that's the immuno from the base pair, and on the right top panel, that's from the amino. So even from the immuno, and based on the location, we can tell for this cone, like it's a GC base pair, typically appears above 12.5 ppm, and 12.2 to 13.5 ppm. And then the AU base pair is a little bit more, even more downfield shift. And then the G appears around 10, and also U appears around 11 to 12 ppm. So this kind of the information can dramatically help you on the assignment of the immuno. So we also can look at the carbon dimension as well. So for the the sugar and the base, there's actually several proton attached to the carbon, and one is the H1 primes on the sugar, then also the adenosine H2 and also H6HA from the pyrimidine and the purine. So I particularly like the the H6HA from the aromatic proton, and this regions each of the nucleotides have one cross peak. And also you can see from the different colors, like guanine, cytosine, they are relatively located at a different location. So this is a carbon proton HSQC is kind of equivalent to the protein, nitrogen HSQC. Basically, each of the cross peak corresponding to one nucleotide, and it can give you the nucleotide level of information. So with this assignment, then we can look at how this how this RNA responding to the ligand or at our nucleotide level. So we can do our titration to add a different compound to the RNA, just to track how the peak move, and so we can to look based on the chemical perturbation to look at where the actual compound bond. And for example, here this is the human telomerase P2AB. The compound is apparently bound to the near the fine nucleotide boards. And we also based on the titration can calculate the binding affinity as well. So this traditional approach works well for the RNA less than 50 nucleotide and with a simple hairpin. But however, if it's the RNA bigger than 50 nucleotide, especially with some little complicated, the tertiary structure like pseudonut and three-way junction, the peak becomes extremely crowded. So for that case, we have several approaches for the large RNA. One is a segmental labeling, and this is a segment text from the Victoria Sousa's lab. And basically what you use is use the T7, and you give the RNA segment 1. Then you have the DNA template will base pair with the RNA, then this particular DNA polymerase will, according to the DNA template, to continue on the segment 1 and to synthesize the RNA. So after it's done, you can add the DNAs to degrade the DNA, then add another DNA template to continue synthesis the segment 3. So then you can decide which region you want to do a labeling and which region you don't want to do the labeling. There's also other approaches, for the segmental labeling, like enzymatic ligation, basically to put two strands together. Also, the position selective labeling over RNA law and the fragment annealing, and it basically to anneal two pieces over two strands of RNA. Then, additionally, there's another way to look at the big RNAs using take advantage of adenosine, and the adenosine H2 is very sensitive to the conformation change of the RNA, and particularly if it's in the certain pattern of in the base pair in the stem, and the H2 will appear at a very unique location. This location is typically very empty. Then we can take advantage of this information to check if it's the stem is forming or lock and form. They look at the bigger RNA. So of course, and another my favorite labeling approach is a furin, and furin is very sensitive and very simple. And the CRS have a very different type of labeling even on the sugar on the base. And this is just an example. You can look at the pseudonet, and it's when it's a single strand from pseudonet, it appears at a different location. Also, it's from a very high order complex appearance at a different location. So you can differentiate and different conformation using furin. So you can even use one step further and put a couple of weights of carbon, and then look at the carbon furin HSQC. So basically, this way is that you look at the RNA complex system, and this can tell you which residue or how many residue is involving with the helix, some is not involving with the helix. So it can tell you to kind of have a rough idea of this conformation of the RNA. So I just want to summarize here, the NMR spectroscopy, especially powered with isotope labeling, is a very powerful tool to look at the RNA structure, dynamic, and binding information. It's a great approach to facilitate RNA-targeted drug discovery. So at the end, I want to thank I'm building my lab. I want to thank my current lab member and also my former lab. Yeah, thank you.
Wonderful. Thank you for the great overview. It's really neat to see, and especially because there's a lot of space for advancement in the field in this area of RNA research. So thank you very much for the overview. One quick question. If somebody were looking to get into RNA research, in your opinion, what would be the best starting point for them out of the common approaches, whether it be chemical or enzymatic, and the labeling patterns currently available?
Yeah, I would say it depends on the size. If the size is less than 150, I would say the chemical synthesis is a straightforward. You don't need to set up a lab and to do it. And otherwise, if it's over 200, the chemical synthesis will be extremely expensive. And I mean, in vitro transcription would be better option.
Okay, so definitely dependent upon the size of
Yeah.
the length of the RNA that you're looking for. And from your perspective, what is needed to further advance the research field in the RNA space currently?
I think so for the RNA-targeted drug discovery, I think what we lack is the structure-function relationship. And basically, that's kind of one of the big hurdle is, because the RNA tend to be very dynamic and have a lot of different conformation, which conformation actually is a structure relevant or a disease relevant is still larger unknown. And if we know which structure is disease relevant, then we can I would say it's a kind of straightforward. You can design it's kind of like a drug or a way to target this particular conformation. But so far, this information is a little bit behind protein study.
Absolutely. Lots of room for advancements for sure. For RNA 3D structure analysis, how many 2D, 3D nosy spectra do you need to collect in general?
Oh, good question. If it's an RNA, it's a small RNA, and I would say it's the D2 nosy, immunonosy. That's probably is good enough for the assignment. However, if it's RNA is I would say bigger than the 30 nucleotide, and then it would require a significant different type of labeling, the nucleotide specific labeling and uniform labeling, and then you have to collect the more advanced like the individual field aided nosy spectrum. So that could be quite a bit. Yeah.
Awesome. Wonderful. Well, thank you very much once again for a wonderful talk. And if anyone has any other questions for Yaqiang or any of our other speakers, please go ahead and put them in the Q&A box.
Yeah. Thank you.
Thank you.